Now from there, from the alpha-ketoglutarate dehydrogenase, you have heard the word dehydrogenase. What is coming to your mind? We are converting NAD positive to what? To NADH. That is the meaning. And what again happen? Decarboxylation occurs. Decarboxylation occur by removing carbon in form of carbon dioxide. So this is decarboxylation, and we are moving from 5 carbon to 4 carbon molecule. So we are converting alpha-ketoglutarate to succinyl-CoA by an enzyme known as alpha-keto dehydrogenase. Also stimulate and inhibit this reaction. ATP does the same thing. So whenever there is high level of ATP starts inhibiting this enzyme. This enzyme in return starts inhibiting the conversion of alpha-ketoglutarate to succinyl-CoA. Now from this, what else do we have? We now have from succinyl-CoA to succinyl-CoA or succinate. So from succinyl-CoA, we now have what? Succinate. And it is done by succinyl-CoA synthetase. Succinyl-CoA synthetase converts succinyl-CoA to succinate by converting GDP to GTP. How? So during the process by which we are converting succinyl-CoA to succinate, what are we doing? What exactly are we doing? We are releasing phosphate group. So this phosphate group will now come, and the GDP will now come and take this phosphate group. And as you take this phosphate group, it now form what? GTP. This DP means diphosphate, this TP means what? Triphosphate. So we have added another molecule. So this succinyl-CoA synthetase converts succinyl-CoA converts succinyl-CoA to succinate, and during this process, CoA is released, coenzyme A is released, and when coenzyme A is released, phosphate is also released, and this phosphate now reacts together with what? GDP to now form GTP. Get ready for my questions. So from the succinate, we now have the fumarate. And it is done by succinate dehydrogenase by taking FAD to what? FADH. This FADH will now go into the matrix of the mitochondria in electron transport chain to produce what? ATP, depending whether ATP level is high or low. When ATP level is low, this FAD is converted to FADH to produce ATP in the electron transport chain. So from fumarate is converted to malate. This is fumarate and this is fumarate, and fumarate is converted to malate by an enzyme known as what? Fumarase. How? This fumarate now take water molecule, and this process is known as what? Hydration. So what is the function of this fumarase? The function of fumarase is to add water molecule, is to hydrate fumarate in order to form what? Malate. So fumarase is hydrating fumarate. Hydration means we are adding water. Dehydration means we are removing water. Carboxylation means we are adding carbon in form of carbon dioxide, and decarboxylation means we are losing carbon dioxide. So malate is now converted to what? Oxaloacetate by an enzyme known as malate dehydrogenase. Malate dehydrogenase convert malate to oxaloacetate by malate dehydrogenase by converting NAD positive to what? To NADH. Now, what is the end product of all these things? So in tricarboxylic acid cycle, which we also known as oxidative phosphorylation, what are we holding? Remember, some textbook will tell you one NADH will hold 3 ATP depending on what you are calculating with. Some textbook will tell you one NADH will hold 2.5 ATP. So it's not depending on what you are given. or what you are using or what you decided to use. So here I decided to use 2.5 ATP. One NADH will give us 2.5 ATP. How? In the electron transport chain. You understand that? Just remember that NADH yield ATP in the electron transport chain, so that when we start discussing about the electron transport chain, you understand that better. So one NADH in the tricarboxylic acid cycle, we produce total of what? 3 NADH, 1 FADH, and 1 GTP. If you go back and count them, you see that your total NADH you are producing are 3. Your FADH is 1, and your GTP is what? 1. And FADH, some textbook will tell you that FADH will yield 2 ATP, and some textbook also tell you FADH will yield 1.5 ATP. So me, I'm using 1.5 ATP. So anyone you feel like using, or anyone they are giving to you. And generally accepted, 1 GTP is equivalent to what? 1 ATP. So the amount of energy one ATP is gonna do, 1 GTP will also do the same thing. So says that in the Krebs cycle, we produce 3 NADH, 1 FADH, and 1 GTP. So now what is in your mind is, if one, so let's go to calculation. If 1 NADH will yield 2.5 ATP, therefore 3 NADH will yield what? X. So when you cross multiply, X times 1 is X, and 3 times 2.5 is 7.5. So therefore 3 NADH is equivalent to what? 7.5. And calculation again, if 1 FADH2 will yield 1.5 ATP, therefore 1 FADH2 is equivalent to what? 1 ATP. And finally 1 GTP is equivalent to what? 1 ATP. So if you add 7.5 plus 1.5 plus 1, your total energy is 10 ATP. So per acetyl-CoA you are producing, how many energy are you producing? You are producing 10 if you are calculating with 2.5. So this now brings to us to our notice that 1 glucose yields 2 pyruvates, and these 2 pyruvates, 1 pyruvate is here, 1 pyruvate is here. This 1 pyruvate will enter and produce its acetyl-CoA, and this pyruvate will enter and produce this acetyl-CoA. So to tell you that 1 molecule of glucose is capable of yielding or producing huge amount of energy, of about 15 to 20 energy, or even 30. So 1 ton of tricarboxylic acid cycle, what is it doing? It's producing 3 NADH. 1 ton is producing 3 NADH, 1 FADH, and 1 GTP. So the net reaction you take home is acetyl-CoA reacts with 3 NAD positive. That's why we have 3 NADH. 1 FADH, that's why we have 1 FADH. 1 FAD. So acetyl-CoA reacts with 3 NAD positive, that's why we have 1 NAD, 3 NADH, and 1 FAD, that's why we have 1 FADH. 1 GDP, that's why we have 1 GTP. 1 pyruvate, that's why we have 1 GTP. 2 molecules of water to yield 2 carbon dioxide, because the end product of tricarboxylic acid cycle is oxaloacetate, which is 4 carbon molecule, while citrate is 6. So when they undergo this process and return back to oxaloacetate, we now have what? 4 carbon. Therefore 2 have undergoes decarbonization and yield 3 molecules of NADH, yield 1 molecule of FADH2, 1 molecule of GTP, 1 molecule of coenzyme A, and 3 hydrogen 3 hydrogen ion that will enter into the mitochondria to produce energy. Don't forget all those things. Just remember that energy is produced in the electron transport chain in the mitochondria. So what are our inhibitors and what are our activators?